Excretory products and their elimination. Ammonia, urea, uric acid, carbon dioxide, water, and ions like sodium ion, potassium ion, chlorine ion, phosphate, sulfate, etc. are accumulated by animals either by metabolic activities or by other means like excess ingestion. These substances totally or partially have to be removed. In this chapter, you will learn the elimination mechanisms of these substances with special emphasis on common nitrogenous wastes. The major forms of nitrogenous wastes like ammonia, urea, and uric acid are excreted by animals. The most toxic form, ammonia, requires large amount of water for its elimination, whereas the least toxic being uric acid, which can be removed with a minimum loss of water. Ammonotylism is the process of excreting ammonia. Many organisms that are ammonotylic in nature are aquatic insects, bony fishes, and aquatic amphibians. As ammonia is readily soluble by diffusion, it is generally excreted across body surfaces or through gill surfaces as ammonium ions. Any significant role is not played by the kidneys for its removal. The production of lesser toxic nitrogenous wastes like urea and uric acid necessitated the terrestrial adaptation for conservation of water. As mammals, many terrestrial amphibians and marine fishes mainly excrete urea. They are called ureotylic animals. In the liver of these animals, ammonia produced by metabolism is converted into urea, which is released into the blood, and by the kidneys it is filtered and excreted out. In the kidney matrix, some amount of urea may be retained in some of these animals to maintain a desired osmolarity. Nitrogenous wastes as uric acid in the form of pellet or paste are excreted with a minimum loss of water by reptiles, birds, land snails, and insects and are called uricotylic animals. A variety of excretory structures are presented by a survey of animal kingdom. These structures are simple tubular forms in most of these invertebrates, while complex tubular organs called kidneys are present in vertebrates. Some of these structures are mentioned here. The excretory structures in Platyhelminthes, rotifers, some annelids, and the cephalochordata amphioxus are protonephridia or flame cells, primarily concerned with ionic and fluid volume regulation, that is, osmoregulation, are protonephridia. In earthworms and other annelids, the tubular excretory structures are nephridia. A fluid and ionic balance is maintained by the nephridia by eliminating nitrogenous wastes. The excretory structures of most of the insects, including cockroaches, are Malpighian tubules. The removal of nitrogenous wastes and osmoregulation is brought about by Malpighian tubules. The excretory functions are performed by the antennal glands or green glands in crustaceans like prawns. Human excretory system. A pair of kidneys, a pair of ureters, a urinary bladder, and a urethra constitute the excretory system in humans. Between the levels of last thoracic and third lumbar vertebra, close to the dorsal inner wall of the abdominal cavity, the kidneys are present, which are reddish-brown bean-shaped structures. In adult human, each kidney measures 10 to 12 centimeters in length, 5 to 7 centimeters in width, 2 to 3 centimeters in thickness, with 120 to 170 grams of an average weight. A notch called hilum is present towards the center of the inner concave surface of the kidney through which ureter, blood vessels, and nerves enter. A broad funnel-shaped space called the renal pelvis is present inner to the hilum with projections called calyces. A tough capsule is present in the outer layer of kidney. Two zones, an outer cortex and an inner medulla, is present inside the kidney. The medulla is divided into medullary pyramids projecting into the calyces. In between the medullary pyramids, the cortex extends as renal columns called columns of Bertini. 1 million complex tubular structures called nephrons, which are the functional units, are present in each kidney. The glomerulus and the renal tubule are the two parts present in each nephron. Glomerulus is a tuft of capillaries formed by a fine branch of renal artery, the afferent arteriole. The efferent arteriole carries away blood from the glomerulus. The renal tubule begins with a Bowman's capsule, a double-walled cup-like structure which encloses the glomerulus. The Malpighian body or renal corpuscle is the glomerulus along with Bowman's capsule. The tubule continues further as proximal convoluted tubule which forms a highly coiled network. The next part of the tubule is a hairpin-shaped Henle's loop which has a descending and an ascending limb. The ascending limb continues as distal convoluted tubule, another highly coiled tubular region. The DCTs of many nephrons open into collecting duct, a straight tube, many of which converge and open into the renal pelvis in the calyces through medullary pyramids. In the cortical region of the kidney, the Malpighian corpuscle, the PCT and DCT of the nephron are situated, while into the medulla dips the Henle's loop. The Henle's loop is too short in majority of nephrons and extends only very little into the medulla. These nephrons are called cortical nephrons. The Henle's loop is very long in some of the nephrons and runs deep into the medulla, and these nephrons are called juxtamedullary nephrons. From the glomerulus, the efferent arteriole emerges and around the renal tubule forms a fine capillary network called the peritubular capillaries. Parallel to the Henle's loop runs a minute vessel of this network forming a U-shaped vasa recta. In cortical nephrons, vasa recta is absent or highly reduced. Urine formation. Glomerular filtration, reabsorption and secretion are the three main processes that involves urine formation which takes place in different parts of the nephron. The filtration of blood is the first step in urine formation and is called glomerular filtration, which is carried out by the glomerulus. The kidneys filter 1100 to 1200 ml of blood per minute on an average, which constitute roughly one-fifth of the blood pumped out in a minute by each ventricle of the heart. Through three layers, the glomerular capillary blood pressure causes filtration of blood. That is, the three layers include the endothelium of glomerular blood vessels, the epithelium of Bowman's capsule, and the basement membrane between these two layers. The podocytes, epithelial cells of Bowman's capsule, are arranged in an intricate manner so as to leave some minute spaces called filtration slits or slit pores. Through these membranes, blood is filtered so finely, except the proteins, almost all the constituents of the plasma pass into the lumen of the Bowman's capsule. Therefore, it is considered as a process of ultrafiltration. The glomerular filtration rate is the amount of the filtrate formed by the kidneys per minute. GFR is approximately 125 ml per minute in a healthy individual, that is 180 liters per day. The kidneys for the regulation of glomerular filtration rate have built-in mechanisms. The juxtaglomerular apparatus carries out by one such efficient mechanism. Formed by cellular modifications, JGA is a special sensitive region in the distal convoluted tubule and the efferent arteriole at the location of their contact. The JG cells can be activated by a fall in GFR to release renin, which can stimulate the glomerular blood flow and thereby the GFR back to normal. By comparing the volume of the filtrate formed per day with that of the urine released suggests that the renal tubules has to reabsorb nearly 99% of the filtrate. This process is called reabsorption. In different segments of nephron, the tubular epithelial cells perform this either by active or passive mechanisms. For example, in the filtrate, substances like glucose, amino acids, sodium ions, etc. are reabsorbed actively, whereas by passive transport, the nitrogenous wastes are absorbed. In the initial segments of the nephron, reabsorption of water also occurs passively. Substances like hydrogen ion, potassium ion, and ammonia are secreted by the tubular cells into the filtrate during urine formation. An important step in urine formation also is tubular secretion, as it helps in the maintenance of ionic and acid-base balance of body fluids. Function of the tubules. Proximal convoluted tubule. A simple cuboidal brush border epithelium lines PCT, which increases the surface area for reabsorption. This segment reabsorbs nearly all of the essential nutrients and 70 to 80% of the electrolytes and water. The pH and ionic balance of the body fluids is also maintained by PCT by selective secretion of hydrogen ions, ammonia, and potassium ions into the filtrate and by absorption of bicarbonate ions from it. Henley's loop. In this segment, reabsorption is minimum. However, in the maintenance of high osmolarity of medullary interstitial fluid, this region plays a significant role. In Henley's loop, the descending limb is permeable to water but almost impermeable to electrolytes. As it moves down, this concentrates the filtrate. The ascending limb allows transport of electrolytes actively or passively but is impermeable to water. Therefore, it gets diluted as the concentrated filtrate passes upwards due to the passage of electrolytes to the medullary fluid. Distal convoluted tubule. In this segment, conditional reabsorption of sodium ions and water takes place. To maintain the pH and sodium-potassium balance in blood, DCT is also capable of reabsorption of bicarbonate ions and selective secretion of hydrogen and potassium ions and ammonia. Collecting duct. From the cortex of the kidney, this long duct extends to the inner parts of the medulla. From this region, large amounts of water could be reabsorbed to produce a concentrated urine. This segment, to keep up the osmolarity, allows passage of small amounts of urea into the medullary interstitium. By the selective secretion of hydrogen ion and potassium ions, it also plays a role in the maintenance of pH and ionic balance of blood. Mechanism of concentration of the filtrate. Mammals have the ability to produce a concentrated urine, as the Henley's loop and the vasa recta play a significant role in this. The flow of filtrate is in opposite direction in the two limbs of Henley's loop and hence forms the counter current. Through the two limbs of vasa recta, the flow of blood is also in a counter current pattern. Towards the inner medullary interstitium, the proximity between the Henley's loop and vasa recta and also the counter current in them help in maintaining and increasing osmolarity, that is from 300 milliosmoles per liter in the cortex to about 1200 milliosmoles per liter in the inner medulla. Mainly sodium chloride and urea cause this gradient. By the ascending limb of Henley's loop, NaCl is transported, which is exchanged with the descending limb of vasa recta. Through the ascending portion of vasa recta, sodium chloride is returned to the interstitium. Similarly, through the thin segment of the ascending limb of the Henle's loop, small amounts of urea enter and the collecting tubule transports it back to the interstitium. This transport of substances facilitated by the special arrangement of Henle's loop and vasa recta is called the countercurrent mechanism. In the medullary interstitium, this mechanism helps to maintain a concentration gradient. An easy passage of water from the collecting tubule is brought about by the presence of such interstitial gradient, thereby concentrating the filtrate. Urine nearly 4 times concentrated than the initial filtrate can be produced by the human kidneys. Regulation of kidney function. The hormonal feedback mechanisms involving the hypothalamus, JGA, and to a certain extent the heart is efficiently monitored and regulated by the functioning of the kidneys. Changes in blood volume, body fluid volume, and ionic concentration osmoreceptors in the body are activated. These receptors can be activated by the excessive loss of fluid from the body, which stimulate the hypothalamus to release antidiuretic hormone, or vasopressin from the neurohypophysis. Water reabsorption from latter parts of the tubule is facilitated by ADH, thereby preventing diuresis. The osmoreceptors can be switched off by increase in body fluid volume, and they suppress the ADH release to complete the feedback. By its constrictory effects on blood vessels, ADH can also affect the kidney function, which causes an increase in blood pressure. The glomerular blood flow and thereby the GFR can increase with an increase in blood pressure. The complex regulatory role is played by the JGA. The JG cells can be activated by a fall in glomerular blood flow or glomerular blood pressure GFR to release renin, which converts angiotensinogen in blood to angiotensin 1 and further to angiotensin 2. The glomerular blood pressure and thereby GFR are increased by angiotensin 2, which is a powerful vasoconstrictor. The adrenal cortex is activated by angiotensin 2 to release aldosterone. Sodium ion and water are reabsorbed by aldosterone from the distal parts of the tubule, and this also leads to an increase in blood pressure and GFR. Generally, the complex mechanism is known as the renin-angiotensin mechanism. Atrial natriuretic factor can be released by an increase in blood flow to the atria of the heart. Vasodilation can be caused by the ANF, and thereby the blood pressure decreases. On the renin-angiotensin mechanism, ANF mechanism therefore acts as a check. Micturition. The nephrons form urine and is ultimately carried to the urinary bladder, where it is stored till the central nervous system gives a voluntary signal. This signal is initiated as of the urinary bladder stretches and gets filled with urine. The stretch receptors present on the walls of the bladder in response send signals to the CNS. To initiate the contraction of smooth muscles of the bladder, the CNS passes on motor messages and simultaneously relaxes the urethral sphincter, causing the release of urine. The process by which urine is released is called micturition, and the neural mechanisms which cause it is called micturition reflux. On an average, 1 to 1.5 liters of urine per day is excreted by an adult human. The urine formed is a watery fluid that is light yellow in color and is slightly acidic with a characteristic odor. On an average, per day 25 to 30 grams of urea is excreted out. The characteristics of urine is affected by various conditions. Many metabolic disorders as well as malfunctioning of the kidney can be clinically diagnosed by the analysis of urine. For example, in urine, presence of glucose and ketone bodies are indicative of diabetes mellitus. Role of other organs in excretion. Other than kidneys, the elimination of excretory waste also takes place in lungs, liver, and skin. Large amounts of carbon dioxide and significant amounts of water are removed by our lungs every day. The largest gland in our body, the liver, secretes bile-containing substances like bilirubin, biliverdin, cholesterol, degraded steroid hormones, drugs, and vitamins. Ultimately, along with digestive waste, most of these substances pass out. Through their secretions, the sweat and sebaceous glands in the skin can eliminate certain substances. The sweat glands produce sweat, which is a watery fluid containing sodium chloride, small amounts of urea, lactic acid, etc. On the body surface through the primary function of sweat is to facilitate the cooling effect. It also helps in elimination of some waste mentioned above. Through sebum, sebaceous glands eliminate certain substances like sterols, hydrocarbons, and waxes. A protective oily covering is provided by this secretion for the skin. Do you know that through saliva, too small amounts of nitrogenous waste can be eliminated? Disorders of the excretory system. Accumulation of urea in blood is caused by the malfunctioning of kidneys, and this condition is called uremia, which is extremely harmful and may lead to kidney failure. Urea can be removed by a process called hemodialysis in such patients. From the convenient artery, blood drained is pumped out into dialyzing unit after adding an anticoagulant such as heparin. The unit, surrounded by a fluid, contains a coiled cellophane tube with the fluid having the same composition as that of plasma except the nitrogenous wastes. The porous cellophane membrane of the tube, based on concentration gradient, allows the passage of molecules. In the dialyzing fluid, the nitrogenous wastes are absent and these substances freely move out, thereby clearing the blood. Through a vein, the cleared blood is pumped back to the body after adding antiheparin to it. All over the world, this method is a boon for thousands of uremic patients. In the correction of acute renal failures, kidney transplantations is the ultimate method. From a donor who is preferably a close relative, a functioning kidney is used in transplantation to minimize its chances of rejection by the immune system of the host. The success rate of such a complicated technique have increased with modern clinical procedures. Renal calculi. Within the kidney, stones or insoluble mass of crystallized salts are formed. Glomerulonephritis, inflammation of glomeruli of kidney. Thank you so much for watching. Please share this video with your friends. Leave your comments below and please subscribe to my channel for more content. Click that bell notification that would let you know when a new video is uploaded. Thank you so much for watching. Once again, please subscribe to my channel for more content. For more videos, please check the description box down below.